Lesson 2, Lipids. Lipids are biological macromolecules and they are polymer which composed of carbon, oxygen and hydrogen. So lipids have carbon, oxygen and hydrogen. Lipids dissolve in nonpolar solvent such as carbon tetrachloride and benzene, but they don't dissolve in polar solvents such as water. So lipids dissolve in benzene and carbon tetrachloride because they are nonpolar solvents, and they don't dissolve in water because water is a polar solvent. Okay, the monomer of lipid, the structure of lipid. Lipid consists of three fatty acids and glycerol. So the monomer of lipids is three fatty acids and glycerol, and glycerol is an alcohol which is having three hydroxyl group. Okay, and that's the structure of lipids. Lipids, the monomer of lipids, three fatty acids and glycerol, and as we said, glycerol is alcohol, and glycerol has three hydroxyl group, 3OH. Okay, classification of lipids. Lipids have three types, which are simple lipids, complex lipids, and derivative lipids. Let's start with the simple lipids. Simple lipids, they form from the reaction of fatty acids in alcohol, and lipids, simple lipids have three types according to what? According to the saturation of fatty acids and the type of alcohol. So simple lipids has or have three types. Simple lipids have three types, and according to what? According to the saturation of fatty acid and the type of alcohol. The word saturation means what? Means the amount of hydrogen. The more amount of hydrogen we have, the more the compound becomes saturated. So more saturation or more saturated means it becomes solid, and means that the compound is having more hydrogen. So more hydrogen equal more saturation, more hydrogen more saturation, less hydrogen less saturation or unsaturated, and the compound will be liquid. So the solid compound, it means that it's a saturated compound or saturated fatty acid, and it means that it has a large amount of hydrogen. Okay, so the simple lipids is classified into three types according to the saturation of fatty acids and the type of alcohol, into three types, which are oils, fats, and wax. Okay, oil, fats, and wax, and they are the three types of simple lipids. Oil is liquid, and it means that it has less amount of hydrogen, and it means that the fatty acid is unsaturated. So oils, liquids, and they consist of unsaturated fatty acids, or it's having less amount of hydrogen, and the glycerol. We can find the oils on the feathers of aquatic birds, the birds which lives beside water. Why? Because oil will prevent the water penetration. Water can't get inside the feathers of this bird, so it can move freely. Because if water is inside the feathers of birds, the bird can't move. Okay, fats. Fats are solids, so they consist of saturated fatty acids, and it means that the fatty acid has a large amount of hydrogen. So fats are solid, so the fatty acid is saturated, and also glycerol. We can find fats under the skin of some animals, like the animals which lives in the two poles, like polar bear. Why? Because they have snow around them. They have very low temperature around them, so they need to keep the temperature of their body inside their body. So the fats under the skin prevent the losing of temperature, or keep the temperature inside their body. Okay, then we have wax. Wax is also solids, but wax consists of high molecular weight fatty acids and monohydric alcohol. Monohydric means that the alcohol is having only one hydroxyl group, not three. So wax consists of high molecular weight fatty acid and monohydric alcohol. We can find wax covering the plant leaves, especially the plant in desert, because in desert the amount of water is very low. So the plant needs to keep the water inside its body. It needs to not to lose this water. So the layer of wax that is covering the leaf reduces the losing of water by transpiration process. So here we have three types of simple lipids: oil, fats, and wax. Oils are liquid, fats solid, wax solid. Oil, unsaturated fatty acid and glycerol. Fats, saturated fatty acid and the glycerol, wax, high molecular weight fatty acid, and the monohydric alcohol. Okay, oil covering the feathers of aquatic birds, fats under the skin of some animals, wax covering the plant leaves. Then the second type of lipids, as we said before, that lipids classification of lipids, they are three types: simple and complex and lipid derivative or derivative lipids. We finished the simple lipids, now we will have the complex lipid. Complex lipids, they formed from oxygen and hydrogen and sulfur and also phosphorus. They have phosphorus element. Complex lipid like what? Like phospholipids. Phospholipids, we can find it in the membrane of plant cell and animal cell. The plasma membrane of plant cell and the animal cells is having phospholipids in its structure. Okay, the structure of phospholipid. We said that the lipids consist of three fatty acids and the glycerol. The structure of phospholipid is the same, but here we have only two fatty acid and the glycerol, and the fatty acid number three is replaced by choline group and phosphate group. So the structure of phospholipid is like the structure of fats or any lipid, but phosphate group and the choline group replaces the third fatty acid. So it has only two fatty acids. Okay, then we have the lipid derivative. The word derivative means converted or they are derived from complex and simple lipids by hydrolysis. The word hydrolysis means that adding water to complex lipid um, and simple lipid, and after adding water they will convert it to another type of lipid which is lipid derivative or derivative lipids. So derivative lipids is derived from complex and simple lipids by a process called hydrolysis, and it means adding water. Okay, lipid derivatives like what? Like cholesterol, like hormones, like steroids. Okay, so the three types of lipids: simple, complex, derivative. Simple, oil, fats, wax. Complex, phospholipid, and we can find a phospholipid in the plasma membrane, and it has only two fatty acids. Lipid derivative, they are derived from simple and complex by hydrolysis, and examples of lipid derivative or derivative lipids: cholesterol, hormones, and steroids. Here we have the structure of phospholipids. We said it has only two fatty acids. Here we have number one, fatty acid 2, but the fatty acid number 3 is replaced by phosphate group and choline group. So it has phosphorus and it has also nitrogen. So phospholipids has phosphorus and the nitrogen, besides carbon and hydrogen and oxygen. So two fatty acids, and the third fatty acid is replaced by phosphate group and choline group, and we can find the phospholipid in the plasma membrane of plant cell and animal cell. Okay, importance of phospholipids. Phospholipid is also a source of energy like carbohydrates, but carbohydrates give us energy faster than lipids. Okay, and lipids give us energy more than carbohydrates. So carbohydrates is faster, but lipids give us more energy. Like if we are having 100 gram carbohydrates and 100 gram lipids, the 100 gram lipids produce more energy than the 100 gram of carbohydrates, and our body begins to release the energy from lipids when it runs from carbohydrates, when there is no carbohydrates inside our bodies. So our body starts to burn the lipids that is stored in the body to get energy. Number two is the main component of cell membrane. As we said, phospholipid, we can find it in the structure of plasma membrane or cell membrane. Okay, number three, they are in the structure of organic compounds in the living cells, like 55% of the organic compounds in the cells. So it's in the structure of cells. Number four, some animals like polar bear and seals, they store the lipids, especially fats, under their skin, as we said before, in the sample lipids. Why? To protect them from the low temperature, to keep the temperature inside their bodies. Okay, number five, they work as protective layers. Protective layers like what? Like the wax that is covering the plant leaves to reduce losing water. And also, we can find lipids in some animals like steroids and also human like steroids, and also they work as hormones. How to detect lipids? How to know if there is a lipid in a sample or in food or not? By using a reagent called sodium 4. If we add sodium 4 to the food or any sample, the color of sodium 4 will convert into red. If the color changes into red, then we have lipids. If it didn't change into red, then there is no lipids.